Our next speaker is Daniela Zarnescu. Daniela is an expert in RNA processing and transport. She has done some very important work on both the physiology and pathophysiology of RNA biology. I'd just like to mention that she graduated from Bucharest University, which happens to be in Romania, in case you didn't know, and which is just north of the Danube before it enters into the Black Sea. Okay, Daniela, it's all yours.
Thank you so much, Bob. Thank you for having me, and it's been a pleasure to listen to the earlier talks. And okay, let me share my screen. Can you see my screen okay?
Yes.
It's just a little slow. Okay, and can you hear me okay, or do I need to change?
Nope, you're fine.
Okay, so I have a really long title, but what I really want to tell you about today is how TDP-43 proteinopathy alters the translation of specific mRNAs in motor neurons. So the advantage of speaking a little later is that earlier speaker, including Magda, have done a really nice job of highlighting the fact that TDP-43 is a really important RNA binding protein in ALS and related neurodegenerative disorders. And without going into details, I just want to highlight the fact that it can bind a number of RNAs, many of them having been identified by Magda when she was a postdoc. And TDP-43 forms these different types of aggregates. And importantly, and something that we are exploiting in our modeling of TDP-43 proteinopathy is the fact that 97% of patients exhibit wild-type TDP-43 aggregates and in ALS. And then there are 45% of FTD patients with wild-type TDP-43 aggregates. So many years ago, when mutations in TDP-43 were identified, we decided to do what we know how to do best in my lab, which is to model human disease in fruit flies or Drosophila models. So basically what we do in my lab is primarily use Drosophila models in what we call a fly to human approach. So I know that at least some in the audience might wonder why flies and how could these uncover key mechanisms of the human disease and how can they be utilized to learn anything useful about ALS? Well, first I'd like to point out that there is a high degree of gene conservation, with 70% of human disease genes having a fly ortholog. Although not identical, there are very similar systems. As highlighted in this cartoon over here, flies have a simplified but well organized and pretty well understood central nervous system as well as peripheral nervous system. There are incredibly powerful genetic tools. We can do pretty much whatever we want. We can knock down genes, we can overexpress genes, we can do that in a cell type specific manner. We can even add a temporal component if we want. We can even look at motor neuron glia interaction using different expression systems. Other advantages include a short generation time. So pretty much in 10 to 14 days, we can get a new generation that we can study. And there is a relative low cost compared to other model systems. So how can one make a model of ALS in flies, and how would we know that it's a good model? The model that we made is based on overexpression of human TDP-43. And although I know there are a number of caveats associated with overexpression, I hope to convince you that our fly to human approach has enabled us to identify critical pathways important in the human disease. So overexpression of human TDP-43 in motor neurons, and I would say both wild-type TDP-43 as a proxy for those 97% of ALS patients with wild-type TDP aggregates, as well as overexpression of mutant TDP-43 with the exact mutations found in human patients, causes a number of phenotypes that recapitulate the human disease. And this includes neuronal degeneration or neuronal loss, as indicated here by the loss of GFP nuclei, morphological as well as functional abnormalities in the neuromuscular junction, reduction in lifespan compared to control flies that can live up to 100 days in the lab where they're nicely treated and well fed, wild-type TDP-43 as well as mutant TDP-43 cause a significant reduction in lifespan. And as a model of ALS, it better has some locomotor function deficits. And we can look at locomotor function in a number of ways using a variety of assays. Perhaps our favorite assay is called larval turning, and I'm just going to illustrate it here. So here is an animal expressing wild-type TDP-43 in motor neurons. If we turn it belly up, you can then take a timer and measure the time it takes for the animal to turn back to its normal position and start moving. So this is a very simple yet quantifiable assay. And even if you don't look like me at fruit flies on a daily basis, hope we can appreciate that this animal is dragging its posterior end as an indication of neural dysfunction and degeneration. So I'm going to start this movie again. And then to the right, I'm going to start a movie that shows you an animal expressing the same human TDP-43 in motor neurons that was treated with a neuroprotective compound. And I hope you can appreciate that this guy has already turned and has made the first forward movement. Okay. So using this model, we are taking really a number of approaches to figure out the molecular mechanisms that underlie this human disease. We're interested in uncovering RNA processing defects in motor neurons as well as in glia. We are performing a number of genetic screens, whether they're candidate or unbiased genetic screens. We're also beginning to look at motor neuron glia communication in vivo. We've been doing a number of drug screens and have identified a number of candidate small molecules, whether they're FDA-approved drugs or novel small molecules that improve locomotor function in our model, as well as in patient-derived motor neurons in collaboration with a number of colleagues in the field. And we're also interested in metabolic studies, and some of our findings published last year are actually very much in line with Bob's work that he described a little earlier today. So Magda already highlighted the fact that TDP-43 is an RNA binding protein involved in a number of RNA processing steps, with the majority of TDP-43 residing in the nucleus, where it associates with RNA targets and it regulates transcription and splicing. But TDP-43 can also be found in the cytoplasm and the axon, as well as at synaptic sites. So evidence exists that in disease, TDP-43 proteinopathy acts through a combination of loss of function as well as gain of function mechanisms. And one of the hypotheses that explains some of the pathomechanisms associated with TDP-43 is called the ribostasis hypothesis, put forth by Ramaswamy, Paul Taylor, and Roy Parker several years ago. And this hypothesis really states that under stress or during aging, in the context of specific mutations, TDP-43 exits the nucleus and goes into the cytoplasm, where it associates with a number of RNA binding proteins, as well as RNA, specific mRNAs. And as a consequence, some of these mRNAs and some of these proteins could become trapped or sequestered. And ultimately, that causes the depletion of specific mRNAs from the translating ribosomes. So essentially, it alters the ribosome association, and that can have consequences on translation or protein synthesis. And in my lab, we've been interested in identifying these mRNAs as well as these RNA binding proteins and specific consequences on translation in motor neurons using our fly models. Additional evidence that TDP-43 may be regulating translation comes from work from a previous graduate student in the lab, Alyssa Coyne, who showed that in control animals, the endogenous fly TDP-43, as well as overexpressed human wild type and mutant TDP-43 associate both with translating ribosomes, as well as non-translating RNA protein complexes. And this is somewhat similar to known translational regulators such as FMRP and poly-A binding protein. So many years ago, starting with Alisa as a student in the lab, we began to identify mRNA targets through a candidate approach. And since this work has been published, I'm just going to highlight some of the key discoveries. But first, I'd like to take a bit of time to explain to you the neuromuscular system that we're using here. So one of those larvae that I showed you has a locomotor defect when TDP-43 is expressed in motor neurons, can be dissected out, and we can do cell biology and biochemistry on the motor neurons located here in the ventral cord, axons, as well as we can examine the neuromuscular junctions formed by these motor neurons as they form contacts onto the body wall muscles. So one of our first RNA targets encodes a protein called FUCH in flies. In a normal motor neurons, you can see its level of expression in the motor neurons lying here at the midline, as well as in the axons. And in the context of TDP-43 proteinopathy, we found an increased expression of FUCH protein. However, when we go down the axon and look at the neuromuscular junction, there is a reduction in the level of this protein that you can also see quantified here by Western blots. So more protein in the motor neuron cell bodies, less protein at the neuromuscular junction. We've shown that TDP-43 causes a shift in FUCH mRNA association with ribosome consistent with translation inhibition. And as I told you earlier in the talk, although we use flies primarily as a model to discover mechanistic changes, we are very interested in validating and seeing whether these changes are relevant to the human disease. So just as a reminder, FUCH protein is upregulated in motor neurons, cell bodies, and significantly reduced at the NMJ, where it has significant consequences on neuromuscular junction stability and function. And of course, we didn't have the ability to look at neuromuscular junction in the patients, but we were able to look in spinal cords from patients with TDP-43 proteinopathy. And as you can see here, the human homologue of FUCH called MAP1B, or microtubule-associated protein 1B, is also increased in motor neuron cell bodies, suggesting that perhaps we are on the right track in taking this approach where we use the power of the fly to identify targets that we could then validate in patient tissues. In addition to using TDP-43 models, we also use C9 models in flies, and Tanya did a very nice introduction of the C9 mutations and their phenotypic consequences. Using the same system, we this time focused on a target that encodes a molecular chaperone called HSC70 or HSPA8 in humans. And without going into much detail, I want to focus your attention on the neuromuscular junction, where you can see that in controls, this protein forms a donut-like structure. It basically associates with synaptic vesicles in synaptic boutons at places of synaptic vesicle release. However, in the context of C9, this protein is significantly reduced in C9, as I mentioned, but also in TDP-43, where we identified the mRNA for this chaperone, encoding the chaperone as the target of TDP-43 proteinopathy, specifically as a target of translation in the context of TDP-43. So what might be the consequences of dysregulation of this chaperone at the synapse? Using FM143 dye uptake experiments, we showed that in the context of C9, there is a reduction in synaptic vesicle endocytosis compared to controls, and this is rescued by HSC70 for overexpression. We then turned to collaborators, specifically Rita Sattler's lab at the Barrow Neurological Institute, and she showed that in patient-derived motor neurons with C9 hexanucleotide repeat expansions, these protein levels are reduced. However, the RNA is unchanged, consistent with the post-transcriptional modification of this chaperone as discovered in the fly. So this once again showed us that we can use the fly to identify a translational target of TDP-43 that also seems to be altered in a similar fashion in C9 models, with the ultimate consequence of affecting the synaptic vesicle cycle and the functionality and durability of the neuromuscular junction. We like to think of this chaperone as kind of a nice link between ribostasis as well as proteostasis, because it is a translational target of TDP-43, and in turn, it regulates proteostasis by acting as a chaperone of key synaptic vesicle proteins. So we were encouraged by the fact that we can take this approach, that we can validate targets from fly to patient tissues and motor neurons. But we really wanted to identify many more and use unbiased approaches. So, you know, if the ribostasis hypothesis is correct, and I know that many in the field have some questions about it, we should be able to identify RNAs that associate with TDP-43, and at the same time, they do not associate with ribosomes, consistent with translation inhibition. And so to test this, we basically took two approaches in parallel. One is RNA immunoprecipitation from TDP-43 expressed specifically in motor neurons, as well as tagged ribosomal affinity purification or ribotagging using tagged ribosomal proteins expressed specifically in motor neurons. So using this approach, we identified a number of mRNAs associated with TDP-43 in motor neurons. Many of them are overlapping with the targets reported by Magda and others in the field. And I just want to point your attention to the fact that when you look at these volcano plots, our previous target FUCH and some new targets are showing up as enriched mRNAs with TDP-43. What might these encode and what might be the pathways altered using GO-term analysis and bioinformatic tool David? We identified a number of pathways that have to do with proteolysis, oxidative stress response, signaling at the neuromuscular junction, as well as glutamatergic signaling. And when you look at KEGG pathways represented in this wheel form, you see a number of pathways that are relevant to locomotor function, as well as neuromuscular junction development and function highlighted over here. And these include FUCH and these other target DLP that I'm going to tell you more in the next few slides. The second step was to do the ribotagging experiments. So first, we defined the motor neurotranslatome in essentially wild-type neurons. And a number of pathways are highlighted as being important for the normal function of a motor neuron. These include mitochondrial metabolism, proteostasis, as well as metabolic pathways and protein folding. However, when we look at alterations specifically in TDP-43 wild type, one of the key pathways that is emerging is the spliceosome. And this indicates that the spliceosome components are both over-translated as well as under-translated in the context of wild-type TDP-43. Interestingly, in the context of mutant TDP-43, one of the key pathways emerging as down-translated, so down-regulated, is the pentose phosphate pathway that Bob mentioned earlier today. And this actually, I think, is consistent with your findings, Bob. In addition, a number of components of the oxidative phosphorylation complex, so essentially all the majority of the members of the electron transport chain are altered. But again, they show both up translation as down translation, suggesting that some of these changes could be direct consequences of TDP-43 proteinopathy, while other changes could be compensatory. So then finally, we were able to test the ribostasis hypothesis by overlapping the data sets. So the RNA enriched in TDP-43, there are many thousands of them. The RNAs that are depleted on ribosomes on the context of TDP-43, they're in the several hundreds. However, the overlap is rather small. So perhaps the ribostasis hypothesis is true to some extent, and it seems to affect a small number of a few hundred mRNAs that are enriched to TDP-43 and at the same time are depleted from ribosomes. And GO-term analysis shows that these mRNAs that fulfill these criteria are involved in signaling, NMJ development, intracellular trafficking and locomotor function, oxidative processes, as well as various metabolic pathways. So I think I need to hurry up a little bit. So all of these pathways and candidate targets are very interesting. And frankly, it's a challenge to pick your favorite and the one to go after. There are many interesting targets and pathways. So we decided to take a more big picture approach and look at what pathways are important for the normal function of a motor neuron. And one of the emerging pathways is what we call the wingless or the Wnt signaling pathway, which plays an important role in neuronal development as well as synaptic development and plasticity. And one of the mRNAs that was fulfilling those ribostasis hypothesis criteria, enriched in TDP-43 complexes yet depleted on ribosomes, is an mRNA called DLP or DALI-like protein, which is a component of this pathway. And essentially it competes with the wingless or the Wnt pathway in binding this receptor. And without spending too much time here, we did some validation and showed that these mRNA associate with TDP-43. And in the context of mutant TDP-43, this mRNA appears to be insolubilized in urea fractions. So if this is a true target, there should be consequences at the protein level. So going back to our neuromuscular system, we examined the localization of DLP protein at the neuromuscular junction as well as in the ventral cord, and found a small but statistically significant reduction of DLP levels. And in addition to our models of TDP-43 proteinopathy, right now at the validation stage, we added a loss of function model where the endogenous TDP-43 is knocked down specifically in motor neurons, and you can see the quantification here. And interestingly, although through a different mechanism, Shantini-Sokanathan's lab showed several years ago that the mammalian homologue, GPC6, is reduced at the neuromuscular junction in SOD1 mouse models, suggesting that the reduction in this glypican, which plays a role at the neuromuscular junction, is perhaps a more common feature, phenotypic feature of multiple ALS model, albeit through different mechanisms. How about the cell body? To our surprise, when we examined the ventral nerve cords where the cell bodies were, although there was no change in DLP expression in the cell body per se, we found a significant accumulation of DLP protein in what we call the neuropil. This is a very dense area that contains mostly dendrites of the motor neurons, where you see a significant accumulation. The loss of function muta seems to have an intermediate phenotype, suggesting some differences between the loss of function and the gain of function models in this case. We don't really know what they are. We suspect that these are vesicles, and we're very interested in figuring out what membrane compartments might this DLP accumulation associate with. So next, one of the powers of the fly model, if you want, is to use genetic interaction approaches and modulate the level of the mRNA target to see what effect that might have on the TDP-43 proteinopathy phenotypes. And I know this is very busy, but I'm just going to point out that when we overexpress DLP in the context of TDP wild type or mutant, we see a slight but significant improvement in locomotor function. Overexpression of DLP does not seem to have a statistically significant effect in the loss of function model, suggesting that this is an alteration that seems to be physiologically significant to the proteinopathy model rather than the loss of function model. And as expected, DLP RNAi causes a significant enhancement in locomotor deficits as expected, given the DLP overexpression rescue. So then, is this relevant to the human disease? As was the case before with FUCH and MAP1B. We weren't able to look at neuromuscular junctions. We are seeking ways to do that. But interestingly, in spinal cords of patients with wild-type TDP-43 aggregates, GPC6, one of the human homologs of DLP, as you can see here, accumulates in puncta that are reminiscent of those accumulations found in the neuropil in the fly model. So what we're thinking right now is that in the context of a healthy motor neuron, there are deficits. In the context of a healthy motor neuron, DLP is normally expressed at the neuromuscular junction where it regulates aspects of wingless signaling, synaptic development, plasticity. In the context of TDP-43 proteinopathy, there are some interesting and mysterious alterations in the soma and the dendrites. And there is a clear reduction in DLP levels at the NMJ, where we hypothesize has an effect on synaptic plasticity through the wingless signaling pathway. Then these are experiments that we're currently pursuing. But these approaches really are at the whole cell level. They're not as elegant as Magda's experiments with synaptoneurosomes that give you insights at the synapse. So in order to gain insights into synaptic changes, we're taking an approach called Non Canonical Aminoacid Tagging that basically is allowing us to substitute methionine for a different amino acids that binds a mutated tRNA synthetase. And when we express this mutant tRNA synthetase in motor neurons, and we use either biotinylation or fluorescence, we can image and quantify the number of newly synthesized proteins at the neuromuscular junction. And as indicated in these preliminary experiments, there is a reduction in newly synthesized proteins specifically at the neuromuscular junction. And using mass spectrometry and biotinylation, we are in the process of identifying those specific proteins. And then it will be really nice to see how they compare to the targets that came out of the ribotagging. And with that, I just want to summarize. I've showed you that our approach is a fly to human, where we take advantage of the power of the fly model to identify mechanistically what changes in TDP-43 proteinopathy. I showed you that we were able to identify and validate molecular and functional alterations in cytoskeletal proteins, chaperones, mitochondrial components, metabolism. All of these seem to be themes that are occurring in everyone's talk, no matter what approach they take, no matter what model they use. And in future directions, we hope to take advantage of these findings and try to restore the changes that we identify in order to rebalance motor neuron homeostasis. And with that, I'd like to thank you. Sorry, I went a bit over time. And I already mentioned the people who did the work. Thanks to them and the collaborators, patients and their families, as well as the funding agencies. Thank you very much.
Thank you, Daniela. That was wonderful. It's really, it's beautiful work. It really leverages the power of a genetically manipulatable system like the fruit flies. It's really impressive. Again, we're sort of bumping up against time constraints. So there are some questions in the chat box, but I'm holding on to them and I'll get them to you later. Thank you again for a wonderful talk.